Welcome. My name is Liza van Eijk. I'd like to thank you for the opportunity to speak today. Please feel free to reach out by email or on LinkedIn, as unfortunately I cannot be here today in person. Alright, I would like to start with the acknowledgement of country. I acknowledge the Australian Aboriginal and Torres Strait Islander peoples as the first inhabitants of this country, and I pay my respects to the traditional owners and elders, past and present, of the land on which we stand today. Today I will focus on sex differences in the brain, and then I will focus on their link with behavior as well as with gender bias disorders, in particularly autism spectrum disorder. And before I continue, I would like to clarify that we looked into sex differences but not gender differences. That is, we did not focus on whether a person feels or identifies with being male or female. So why is it important to consider biological sex differences? Well, several disorders and diseases have shown sex differences in their prevalence or symptom expression. If we look at this figure here, we see that females are more often diagnosed with anxiety and depressive disorders than males. Vice versa, we see that males are more often diagnosed with autism spectrum disorder and ADHD. Now, investigating sex differences in the brain and or the masculinization process might provide new insights into the skewed sex ratio of these gender bias disorders and their cause. Now, in addition, if we, for example, look at Alzheimer's disease, we see that it shows a more rapid decline in cognition after diagnosis in females in comparison to males, raising the question whether sex differences in the brain make one sex more vulnerable than the other for certain diseases, or a more severe progression of the disease. Now, sex differences may also exist in the efficacy of treatment. However, not until recently, not many studies considered this to look at the outcomes in males and females separately. But nowadays, this is slowly changing for the better due to mandatory changes from funding agencies like the NIH. So why could there be any sex differences in the brain? Well, there are multiple explanations for this. First of all, differences in sex chromosomes, with girls receiving 2 X chromosomes and boys 1 X chromosome and 1 Y chromosome at conception. Now, this genetic difference results in differences in sex hormones. Girls follow the default trajectory. However, in boys, there is a gene on the Y chromosome, that's called the SRY gene, that starts the sequences resulting in the male development. That is, more testosterone and anti-Müllerian hormone to stop the female development. But differences in our environment also may result in differences between males and females. There are, for example, differences in how females versus males are treated and raised due to gender stereotypes. These gender stereotypes do not only relate to toys, but also in the qualities we relate to males and females. For example, the idea that females are more empathetic and are better at taking care of people and children, whereas we have this idea that males are better at building something and, for example, mathematics. And we can see these stereotypes in the gender distribution for several occupations. Now, these environmental differences are important when examining sex differences in the brain, because in reality it is hard to distinguish biological sex versus societal brain and behavioral differences in humans. In addition, we know that genetic and environmental differences interact with one another. Now, when discussing sex differences, it is good to understand that these differences are often on a continuum. And what does that mean? Well, when we look at sex differences, for example for those in height, the distribution for men and women overlaps. So on average, men are taller, but there will be exceptions. So we look at sex differences in the brain on a similar continuum. The largest sex difference in the brain is the difference in brain size. But what about regional differences in the brain? That is, once we adjust for overall brain size. So if males and females would have the same brain size, would there still be any differences? A region particularly of interest is the hippocampus, because this region has a high density of estrogen receptors, which makes it interesting to investigate in relation to sex differences. So the hippocampus is a seahole-shaped subcortical limbic structure and has been related to learning and memory. It can be further divided into subregions. So when we use the software FreeSurfer, we can further divide the hippocampus into 12 regions, as you can see here in the images. We have replicated that male brains are larger than female brains, and we've also shown a sex difference for the hippocampus and the subfields, with larger volumes for males in comparison to females. However, to examine whether there are any sex differences in a specific region, it is really important to consider overall differences in brain size. To take into account differences in size while examining sex differences in the hippocampal subfields, we adjusted results for overall hippocampal volume using 4 different methods, including the allometric approach, the covariate approach, regression, and matching males and females for overall hippocampal volume. We then looked at sex differences in the hippocampal subregions while adjusting for total hippocampal volume. Interestingly, we only found sex differences in 5 of the 12 subfields, particularly in subregions containing white matter or CSF, but not the grey matter regions. And this suggests that sex differences may be related to connectivity and folding differences between males and females. Future research should examine how these sex differences in the hippocampal subfields change across the lifespan, and how these may be related to certain gender-biased diseases that impact the hippocampus, such as Alzheimer's disease. Okay, so for now we looked at structural differences. What about brain function? Our next project focused on regional cerebral blood flow differences. Changes in regional cerebral blood flow have been linked to behavior, such as learning, so anxiety, perceived stress, pain perception, as well as disorders, such as Alzheimer's disease and depressive disorders. Now, cerebral blood flow has found to be 4 to 50% greater in females than in males, but this difference is not found prior to adolescence. The question is whether this difference occurs due to different trajectories during adolescence. Now, a previous study found that pubertal status drives the sex difference in cerebral blood flow. The question is, can we replicate this in a sample of young adolescents? For our study, we looked at to what extent age and pubertal status drive sex differences in cerebral blood flow, and we used the sample of young adolescents scanning twins at the Queensland Brain Institute, including 191 young adolescents that were scanned so far on the free Tesla scanner. And we used a special type of scan to measure cerebral blood flow in a non-invasive way, using arterial spin labeling. So we found overall greater whole brain cerebral blood flow in girls than in boys. And we can see in these plots here that this difference was dependent on age, as you can see on the left, and pubertal status, as we can see on the right. Now we replicated the sex differences in developmental trajectories of cerebral blood flow during adolescence. And this might potentially have implications for behavior and disorders associated with sex differences, in particularly disorders that have shown an increased prevalence during adolescence, such as depressive and anxiety disorders. For the next project, we aim to examine the relationship between global sex differences in the brain with behavior, because sex differences have been found for both the brain as well as behavior. So when we look, for example, at sex differences found for behavior, we can see that males are on average better at, for example, mental rotation tasks in comparison to females, whereas females perform better than males on average on emotion recognition tasks. So the question was really, are these sex differences in the brain related to sex differences observed for behavior? To work around the possible confound that men and women may be treated differently, we looked at masculine versus feminine differences among individuals of the same sex, instead of between men versus women. Now, individuals vary in their genetic predispositions as well as exposure and sensitivity to sex hormones. Some men will develop a more female-like brain, while other men will develop a more exaggerated male-like brain, and conversely for women. Previous work has shown a weak association between brain measures and behavioral measures. However, it wasn't clear whether this relationship was driven by sex differences in brain size. To consider differences in brain size, we used two different approaches to develop the brain masculinity-femininity measure. We used the landmark approach, including subcortical and cortical landmarks to measure brain shape, and we used the vertex-wise approach, measuring thickness and surface area derived from free surfer. Now, using these measures, we trained an algorithm to identify biological sex of a person based on their brain scan measures, and we used brain size as a proxy for comparison. After training, we made sure to test the accuracy of the prediction in an independent sample to avoid overfitting. So as you can see here in this figure, both approaches predicted sex well, but the prediction was often better when brain size was not filtered out. Similar to how we derived the brain measure, we derived a behavioral score by predicting an individual's biological sex based on multiple behavioral measures, including cognition, personality, anxiety, and depressive symptoms. Now next, we looked at the association between these brain and behavioral scores, and we found a weak association within both sexes. However, although we already adjusted for brain size, the brain measures still show the relationship with brain shape. That is, a smaller brain may have a different shape than a larger brain. So to make sure the association wasn't still driven by brain size, we regressed out brain size and then we found no association, suggesting that brain size differences may be driving this association between sex differences in the brain and behavior. In addition, we found that the brain and behavioral differences are moderately heritable, and that genetic and unshared environmental influences explained approximately a similar proportion of the variance. To conclude, we found some evidence for an association between sex differences in the brain and behavior. However, these appear to be driven by brain size. More importantly, we have to note that this is a correlational study. Further, we hypothesized that sex differences in the brain and behavior may be driven by the same masculinization process early in life, in particularly as we found an association between the brain measures with height, which is unlikely due to a causal effect. However, further studies are needed to examine these sex differences across the lifespan and the effects of prenatal versus postnatal hormonal influences on these sex differences. Developing the brain measure of masculine and feminine differences on the continuum allowed us to also test whether brain sex differences may relate to certain gender bias disorders. Now, a classic example of such a gender bias disorder is autism spectrum disorder, because it is about 4 times as common in males as in females. It has also been associated with extreme masculine behavior, as people with autism spectrum disorder score below average on empathizing, but average or above on systematizing. What does this mean? Well, the first dimension, empathizing, consists of 2 different aspects. The first, developing theory of mind, is often delayed in people with autism spectrum disorder. They have trouble reading the thoughts and feelings of their own or others. The second aspect, responding with an appropriate emotion reaction, which is often referred to as showing sympathy, is below average in people with autism spectrum disorder. Now this is the opposite for the other dimension, systematizing. That is the drive to identify rules within a system, to analyze and predict using these rules accordingly, which is often needed in physics, engineering or mathematics. Now this dimension relates to the resistance to change as well as narrow interests and repetitive behavior that's often found in people with autism spectrum disorder, which can lead to superior skills such as excellent eye for detail and the ability to do calculations rapidly. Now this empathizing, systemizing theory is confirmed by using behavioral measures such as empathy and attention to detail tasks. So if we look at the first one, females scored the highest on empathy tasks, whereas males scored in between, and adults with autism spectrum disorder had the lowest score. But this pattern was reversed for the attention to detail tasks, and that suggests extreme masculine behavior in people with autism spectrum disorder, regardless of their sex. This resulted in the idea of the extreme male brain hypothesis proposed by Baron Cohen, suggesting that autism spectrum disorder is related to an exaggerated male-typical brain, possibly explaining the higher male-female ratio. At the time of this study, only one study, including less than 200 individuals, found support for this hypothesis, but they later retracted the results. So here we tested the association between brain differences on the male-female dimension, as well as with autistic traits, using the measure from the previous project. Now note, recently the same lab published evidence for this relationship. For our study, we used the T1 scans of the freely available brai datasets with more than 2,000 individuals. And approximately half of those include autistic brains and the other neurotypical control brains. We extracted the subcortical landmarks for this dataset due to no diffusion scans being available for most of the cohort. To derive the brain masculinity-femininity measure, sex was predicted by training the algorithm on the population samples and then predicting in the abide samples. The brain maleness scores were higher in control males than in control females. And on average, we've seen higher mean brain maleness scores for autistic brains versus control brains, providing evidence for the extreme male brain hypothesis. And we've also related these brain maleness scores with autistic symptoms. However, autistic brains were on average larger than control brains, raising the question whether this is confounded by brain size. After taking into account brain size, no link was found between brain masculinity and autism spectrum disorder diagnosis and symptoms. So, individuals with autism spectrum disorder had slightly more male-typical brains, but this difference is accounted for by males and individuals with autism spectrum disorder having relatively larger brains than females and controls, respectively. To conclude, I would like to summarize the key points of this talk. Sex differences in the brain exist, but male and female brains show large overlap. Remember the height example. Now, based on our findings, we have related sex differences in the brain to sex differences found for behavior. But we only found a weak correlation, which appeared to be driven by both genetic and environmental factors equally. We've also shown a relationship between sex differences in the brain with autistic symptoms and diagnosis. But it's important to note that both the relationship between sex differences in the brain with behavior as well as with autistic spectrum disorder is driven by differences in brain size. And therefore, this shows that it is very important to adjust for brain size when examining sex differences, even if features themselves, such as brain shape, do not necessarily measure brain size. They may be confounded by brain size. So carefully considering the methodology when examining sex differences is necessary to provide a better understanding of whether and how sex differences in the brain relate to behavior and disorders. And this may assist with more accurate and/or earlier diagnosis of conditions that have shown sex differences in their prevalence and/or symptom expression. It may also assist with personalising treatment by improving prediction accuracy of disease progression and improving identification of the most suitable treatment option. Thank you for your attention. Please feel free to get in contact.